All right, thank you for having me. My name is Prasanna Veeratunga, and I work as a postdoctoral associate at the Vanderwaal Lab, and I'm from Baker Institute for Animal Health from Cornell University. And our previous presenter already talked about feline iPSCs. I'm also here going to talk about feline iPSCs, but generated with a slightly different approach. So my talk is about footprint-free feline-induced pluripotent stem cells, and these cells can be successfully differentiated into mesenchymal stromal cells. Sorry. So to begin with, I'd like to give you a brief introduction. So what are the iPSCs? So iPSCs are differentiated cells that have been reprogrammed back into embryonic stem cell-like cells with ectopic expression of defined factors. These iPSCs show properties of self-renewal, as well as they can be differentiated into any type of cell in the body. I'm going to talk here about viral-mediated iPSC generation. So in terms of viral-mediated iPSC generation, there can be integrative viral vectors such as lentiviruses and retroviruses, where these factors integrated with the host genome. And we are going to use integration-free virus vectors like Sendai virus vectors. These vectors do not integrate with the host genome. Therefore, these iPSCs are clinically relevant. So my study has two aims. First is to generate these iPSCs from feline fetal fibroblasts using Sendai virus vectors, and to differentiate these iPSCs into functional mesenchymal stromal cells. So talking about aim 1, so these are my material and method for both the aims. So we used fetal fibroblasts from cat fetuses, and we reprogrammed these fetal fibroblasts using Sendai virus vectors. We used 3 vectors carrying 4 human transcription factors, 4 human genes, and we generated these iPSCs, and we differentiated these iPSCs into MSCs, as well as characterization of both iPSCs and MSCs. In terms of characterization, we check the expression of endogenous pluripotency genes, we check the expression of alkaline phosphatase, as well as we check the differentiation both in vitro and in vivo. So this is the timeline for iPSC generation. Actually, we used commercially available Sendai viral vectors. These kits are very expensive, so you cannot use them directly for reprogramming experiments. So before that, we check if these cells are amenable for Sendai viruses by using this Emerald GFP fluorescent reporter. So we confirmed that these feline fibroblast can be transduced with Sendai viruses using this reporter assay. So this is my timeline for iPSC generation. So we performed the transduction at day 0. Before that, 2 days before, we plated our fibroblast with appropriate cell numbers. After transduction, following day, we changed the media. We keep changing the media for 6 days. On day 7, we plated these transduced cells on inactivated SNL feeders. Following day, we switched to iPSC medium. Then we keep changing the medium until we see emerging colonies. And when colonies emerge, when they are ready to transfer, we again transfer to inactivated SNL feeders. So these are our generation of iPSC colonies. So this is day 0. This is day 0 at the transduction. So I think there's some problem with the PowerPoint slide. So this is day 18. We saw mesenchymal to epithelial-like transduction, epithelial-like transformation. At around day 20 to 24, we saw emerging colonies. At around day 28, these colonies were ready to transfer. So these are the images of established iPSC colony. You can see the typical characteristic dome-shaped colony with defined borders. And if you can observe this white arrow, we can see cells with high nuclear to cytoplasmic ratio. And feline iPSCs has already been generated in 2019, as our previous presenter mentioned. they have been generated, but that was using retroviral vectors, which are integrating vectors. And our iPSCs, in terms of morphology, they were comparable to these iPSCs which has been described before. So on characterization of these feline iPSCs, we first check the expression of these endogenous feline pluripotent genes, feline SOX2, feline NANOG, and feline OCT4. You can see at around passage 3, we could see the expression of feline NANOG and OCT4 initially. Then at around passage 6, we could see the expression of feline SOX2. And these endogenous pluripotent factors, they continue to express all the throughout the passage in time. So even at P15 and P25, we could see the continuous expression of these pluripotent genes. Talking about transgenes or human factors, as the passage number increases, you can see the decrease of this expression of human transcription factors. Interesting thing is at P6, beyond P6, we couldn't detect any of the human transgenes. In terms of Sendai virus, we could see the level of Sendai virus replication decreased with the increase in passage number, and they eventually lost from the host cell. And further on characterization, they were positive for alkaline phosphatase, and we did the karyotype analysis, and we could see normal haploid chromosome number, and they differentiated into embryonic bodies in vitro, which contain markers for endoderm, mesoderm, and ectoderm. And upon differentiating into embryonic bodies, the embryonic bodies lost the expression of endogenous pluripotent genes compared to their parental iPSC lines. And importantly, we try to generate in vivo teratomas using these iPSCs. We injected these iPSCs into hind leg of these immunodeficient mice. At around 5 weeks post-injection, we could see emerging teratomas. This is the harvested teratomas at around 8 weeks post-injection. And we did this analysis with the H&E stain. And interesting thing is we expected to see differentiated tissue in teratomas. But instead, what we saw was like monomorphic population of like cells, like this represented like germ cell-like tumor. This is quite interesting because these are Sendai virus-derived iPSCs, not retroviral or lentiviral-derived iPSCs, which usually generate teratomas in vivo. But the thing is, this is the first report of feline iPSCs using Sendai viral vectors and generation of tumors in mouse. So further studies are needed to explain this outcome. But we checked the expression of feline proto-oncogenes. I haven't included our results here, but we see the elevated level of feline proto-oncogenes like c-MYC and KLF4 in these tumors. For the M2, we try to differentiate these iPSCs into functional MSCs. The thing is, we can harvest these MSCs from tissues, from bone marrow, from adipose, but if we have an established protocol to generate iPSC-derived MSCs, we can get fairly homogeneous MSC population, and that's the one name. In addition to that, we just want to validate the authenticity of these iPSCs, because in downstream we are planning to use these iPSCs for other purposes, so we differentiated them into MSCs. So for the differentiation, we use this traditional embryonic body differentiation assay. You might all know about this assay, but this failed. And then we try to use direct differentiation with this TGF-beta inhibitor. There's validated protocol for human and mouse iPSC-derived MSC generation. That also failed. Then we used another protocol. This protocol has been validated for equine iPSCs, which is a serial passaging of these iPSCs with the presence of ALK inhibitor. By using this protocol, we could generate MSC or MSC-like cells. On characterization of these MSCs, we could see the expression of MSC-specific surface markers, CD29 and all the other accepted MSC-specific markers, both at a transcriptional and translational level. And they were also differentiated into fat, bone, and cartilage in vitro. And this is where I am with respect to this experiment. So we have feline iPSCs, which are well characterized, and we have differentiated iPSC-generated MSCs, which are also well characterized. And we are going to plan this, we are going to use these MSCs for regenerative medicine and immunomodulatory studies. And in the long run, we are going to use these iPSCs to generate feline intestinal organoids to use as in vitro model to support feline coronavirus infection. To conclude, I'd like to thank our PI, Galinda van der Waal, and our research support specialist, Becky Harmon, and also all our lab members. And feel free to ask any questions if you have, and thank you for your time.
Thank you.
Great presentation, thank you. So, 2 questions. Just to clarify the culture conditions, so you use basic FGF in a LIF in the media? That was one question.
Yeah.
And so, if you can tell us the amounts that you put in there.
Yeah, we use feline LIF and human BFGF. So talking about iPSCs, they can be either naive or prime. So I'm not going to say my iPSCs are naive, but what I can say is they LIF dependent, but we add BFGF to support their proliferation. To say that they are naive, more experiments are needed.
What is the concentration of the LIF?
I can't remember at the moment.
So one more question here. So thank you for sharing the histopath images of that tumor, that teratoma, which looks more like a potentially teratocarcinoma rather than a teratoma. So 2 questions. A, how many times did you repeat that, and whether that finding was repeatable? And then how concerned are you about those cells if they're making teratocarcinomas as opposed to, differentiating as you'd expect them to do?
What was the first question?
Was that repeatable? How many times did you do that teratoma?
Okay, so we generated 4 iPSC lines, and we injected 4 of them, but characterization was done on 2 of them. But all these four lines, they generated these tumors. So I can't explain what's happening actually, but further studies are needed. But one thing is, we use the MOIs recommended by the manufacturer, and maybe they are low MOIs, and that's why we had to wait very long time to see the iPSC generation. So that don't know whether that has an effect as well. But in human and in mouse, iPSC is generated from retroviral, antiviral. They show this kind of teratoma generation rather than differentiated tissues. But yeah, I don't know exactly what's the reason behind that.
Last question.
Hi, thank you very much for your interesting result. In your Gale image, you said the footprint was disappeared, but
Yeah.
As far as I remember, maybe the top one, CMYC, was appeared again. So I wonder if you tried different method than RT-PCR, like a qPCR or something else.
Yeah, actually we have validated all this data with qPCR as well, but I included only RT-PCR data here. But we have evaluated these iPSCs at higher passages as well. So they are Sendai virus free and transgenes free.
Thank you so much.
Thank you.